everybody, my name is Deichmann.
Hello everybody, I'm Ishamel. In this video, we are going to present to you our ongoing work about characterization of nutrient imbalance symptoms in selected wheat and barley genotypes.
Plants need 14 essential nutrients, 6 macro and 8 micronutrients. Nutrient deficiencies may cause high yield losses in agriculture, and at the same time, too high concentrations of distinct elements in the soil even may have a toxic effect on the plant. High fertilizer input has a negative environmental impact. Volatile nitrogen compounds such as ammonia, NOx, or nitrous oxide, which may be caused by high ammonium fertilization or anaerobic conditions in the soil, contribute to air pollution and greenhouse gas emission. Nitrate may be leached and pollute the groundwater, as already happened in many areas and not only those with high livestock. Phosphate eutrophication threatens the ecological balance in rivers, lakes, and oceans. Furthermore, unnecessary fertilization has a negative impact on the farmer's economic balance sheet. So there is a need for optimized fertilization in the interest of farmers as well as the environment. The inhomogeneity of soils often makes precise fertilization based on punctual soil samples impossible. Usually, commercially available fertilization systems with optical sensors estimate chlorophyll content and biomass of the crop plants and regulate nitrogen fertilization according to this estimation. However, a low chlorophyll content of a leaf may also be caused by stresses other than nitrogen deficiency, which these systems do not consider. The result can be an unnecessary fertilizer application with all known negative consequences. Therefore, a better detection of nutrient imbalances of crop plants is necessary. Deep machine learning offers the opportunity of developing sensor systems of much higher accuracy. But for training of a convolutional neural network, reliable and exhaustive data sets are necessary. And later on, a tool to validate the quality of the sensor detection in the field. There is a need of crop plants with defined nutrient imbalances to serve as a template for pattern recognition of the caused symptoms, and to obtain reference transcripts for ground truthing of sensor detection on site. In our approach, we grew genetically diverse wheat and barley genotypes under the controlled conditions of a hydroponic system. Seedlings were planted into nutrient solutions of defined composition. Except for the nutrient sufficient control, in each treatment, one of the essential elements was mildly deficient or one element had a toxic concentration, as can be found in natural acidic soils. The experimental setup was a randomized block design with 3 repetitions. One run took 4 to 5 weeks. The period was chosen so that all genotypes remained in the same vegetation stage to keep the phenotypic parameters comparable. After this time, the plants looked like this. They were still in a state before ear shifting. Fertilization would still have a high impact on the yield. Already on the first glimpse, not all treatments develop homogeneously. Compared to control, the growth impairment of plants exposed to aluminium toxicity gets obvious. Also, calcium deficiency causes strong growth depression, whereas, for instance, iron deficient plants do not seem to be affected in the same way. Different nutrient imbalances have different consequences with respect to the growth, but growth depression is not the only symptom. There are many others, like necrotic leaf tips and stripe pattern on the leaves, such as intercostal chlorosis. To document all these patterns, we took lots of RGB images during the whole experimental period, more than 75,000 up to now. Thus, we recorded nutrient imbalance symptoms of 10 genotypes, 5 winter wheat varieties, 5 barley varieties, and of 16 different nutrient imbalances during several weeks of plant development. These data sets are going to serve for training of a convolutional neural network on symptom pattern recognition. Besides RGB imaging, we also evaluated other non-invasive detection methods. Spectral reflectance measurements with so-called polypen handheld resulted in diverse commonly used indices like, for instance, NVDI, NPCI, and PRI. We also generated hyperspectral images of all genotypes and nearly all treatments. But due to high costs and efforts, we could not conduct these measurements as comprehensively as the RGB imaging. The obtained data are still in process. Also, for later processing, we took samples of plant material for transcript analysis to study gene response to nutrient imbalances. Thus, we hope to obtain reference transcriptomes for later ground truthing of sensor detection in the field with high accuracy. Now the methods are getting highly invasive. Of course, we also collected data of important growth parameters to make out differences between the genotypes in nutrient efficiencies and toxicity tolerance. That means the end of each experiment. We measured shoot and root length, number of tillers, fresh weights and dry weights, and after measurements we stored all the dried plant material for later nutrient analysis. So overall, we collected lots of data to characterize nutrient imbalance symptoms of the selected barley and winter wheat genotypes comprehensively.
Welcome to the result part. This figure shows a heat map generated using p-values collected from ANOVA results. The x-axis shows the nutrient imbalance treatments and the y-axis shows the wheat genotypes morphological traits. The first column indicates the effect of nutrient imbalances on genotypes and the second column shows the effect of each nutrient imbalance on these morphological traits compared to the control. The third column is the genotype by treatment interaction effect on those traits. The X sign indicates a significant variation at p less than 0.05. As you see, the result shows there are a significant variation between the genotypes in these traits, such as shoot and root dry weight variation in response to the nutrient imbalance treatments. There is also a huge effect of the nutrient imbalances on those traits compared to the control. In addition, there is also a genotype by treatment interaction effect in some of the treatments for some traits. The heat map only shows the general overview about the variations, but it does not tell you which genotype performed better in each nutrient imbalance. Therefore, we selected some representative data based on the heat map to see the real genotypic differences and treatment effects. These two figures show shoot dry weight and root dry weight accumulation in different wheat genotypes in response to nutrient deficiency treatments. As you see, sulfur and copper deficiency reduced shoot and root biomass accumulation, which is highlighted in red rectangle. As you see in the photo, Sulfur deficiency resulted young and middle-aged leaf chlorosis compared to the control. Sulfur is a constituent of essential amino acids and involved in the biosynthesis of many compounds. So, its limitation in the medium could affect shoot and root biomass. In addition, the wheat genotype, Julius and especially Greif, accumulated higher root and shoot biomass compared to the others under iron, manganese, and molybdenum deficiency, which is highlighted in blue rectangle. These two genotypes, especially Greif, could contain putative genes for iron, manganese, and molybdenum deficiency tolerance. In the upper figure, the shoot-derived accumulation is reduced in nitrogen, phosphorus, potassium, and calcium deficiency treatments, which is highlighted in red rectangle as reported earlier. These are essential macroelements that plants need in large amounts to grow and complete their life cycle. As you see in the upper photo, there is extensive shoot biomass reduction in calcium deficiency. Calcium deficiency causes necrosis of growing points, and the symptom is very strong in weak transpiring organs. Calcium is essential in maintaining cell structure and membrane integrity. The root length was also reduced in calcium deficiency treatment, which is the lower figure highlighted in red. However, the root length is increased in potassium, manganese, molybdenum, and especially in phosphorus deficiency treatments, which is highlighted in blue in the lower figure. Plants increased root-shoot ratio presumably by translocating resources towards root growth under nutrient deficiency treatments. This could be for foraging response. This is again the heat map for the Barley parental magic lines. As you see, there is treatment effect on the traits and there are variations between the genotypes in the traits in response to the nutrient imbalance treatments. There are also some interaction effects. In the upper figure, while deficiency of macronutrients reduced shoot-dry weight in most genotypes, only nitrogen, calcium, and magnesium deficiency reduced root dry weight, which is highlighted in red in the upper and the lower figure respectively. In contrast, phosphorus deficiency increased the root dry weight accumulation as reported earlier, which is highlighted in blue. The deficiency symptoms are visible in most of our treatments. For example, you can see intercostal sclerosis of young and middle-aged leaves in magnesium deficiency treatments, which is the upper photo. We found significant variations between the genotypes as Ragusa accumulated higher shoot-dry weight compared to the others in most of the macronutrient deficiency treatments. Ragusa also accumulated higher root-dry weight in nitrogen and phosphorus deficiency treatments. Therefore, this genotype could contain candidate genes for some macronutrient deficiency tolerance. NDVI or normalized difference vegetation index is considered as a greenness index, and DVI value is reduced in nitrogen, calcium, magnesium, and sulfur deficiency treatments as highlighted in red in the above figure. NDVI estimate photosynthetic capacity and net primary productivity. Since the shoot biomass is reduced in these deficiency treatments, so it is not a surprise if the NDVI is reduced as well. The lower figure shows the root dry weight for aluminum and manganese toxicity treatments. As you see, the root biomass is reduced in both aluminum and manganese toxicity treatments. The molecular mechanisms of aluminum toxicity are not yet identified. Looking genotypic differences, Barca accumulated higher root dry weight compared to the others in response to manganese toxicity. In conclusion, many of the nutrient imbalance treatments affected most of the morphological traits observed, and there are significant variations between the genotypes in these traits in most of the nutrient imbalance treatments. Our future plan is to do deep neural network training with 75,000 images for symptom pattern recognition. This will be done by Jinhui, who is a PhD student with Professor Gal at the University of Bonn. We are also planning to establish reference transcriptomes for defined nutrient imbalances. These transcriptome analyses could support ground truthing efforts under field conditions, particularly in cases where multiple nutrient imbalances may occur and conventional nutrient analysis of soil and plant extracts fail to accurately diagnose nutrient imbalances. Finally, the identified genotypic differences are also the starting point for future mapping approaches to identify QTLs, potentially genes involved in nutrient use efficiency for the breeding of nutrient use efficient crops. Thank you for watching.